Okay, sorry about that. Okay, so like I was saying, if you have a cut or if you have bent yourself, that is direct damage to the endothelial cells. But apart from that, the recruitment of other cells, or let me just say the recruitment of cells to the site of damage to neutralize the toxins, to get rid of the necrotic tissue. Okay, so it sometimes that cascade of events, you find out that leukocytes can also produce what we call radicals. And these radicals, when they are released at the site of injury, instead of doing good, these radicals are dangerous to the site of injury. Then you find out that these radicals, they contribute to further damage of other cells which are okay. Okay, so these leukocytes do not need to spend much time at the site of damage because they produce oxygen species, and these oxygen species, they are very harmful. Apart from oxygen species, they are going to produce proteolytic enzymes which cause endothelial damage and eventually detachment. Okay, so if you look at diseases such as acute lung injury, I think it's acute lung injury, or should be acute or chronic lung injury. Okay, so when you look at such a disease, you find out that the major cause to damage, or the major cause to progression of that same infection, you find out that it's due to the leukocytes, due to them producing those radical species. So you find out that once they are produced, they lead to further damage. So two ways that you can damage endothelial cells: leukocytic dependent endothelial injury, as well as direct injury. Okay, so if we do not have any questions, we can take a break here. So the first question is saying, stating the first vascular response to injury is, okay. Who wants to take this one? Who's taking this question? Hey, no one. Okay, let's start answering. The first vascular response to injury is A. Is saying slowing of circulation. Is A correct or false?
False.
Okay, that's true. Oh, that's false. Yeah, that's correct. B is saying the first vascular response to injury is venular dilation. Is that true or false?
False.
That's true. C is saying recruitment of the vascular beads. Is that true or false?
False. False.
True.
False. D is saying capillary enlargement. Is that true or false?
False.
Okay, false. E is saying atrial vasoconstriction. Is that true or false?
False.
Okay, so the only correct answer here is B, venular dilation. For an explanation, I have placed an explanation here. So once I send the notes, I trust that someone will go through. Okay, this should be the last question. So the first event in acute inflammation is A, atrial vasodilation. True or false? Okay, no one has it. B, increased permeability. True or false?
False.
False. Okay. C, diapedesis. True or false? Okay, since you don't know this term, I'll answer for you. It's false. So the correct answer here is atrial vasoconstriction. That's the first response of acute inflammation. Remember, 8 to 15 minutes of vasoconstriction. Then after we have that that blood at the site of inflammation, the next thing is going to be vasodilation to slow down the movement of blood. Okay, so remember the two events of yeah, I'm sure you know what I'm talking about. So the answer is there as well in case someone is looking for reference. Okay. So cellular events. We've talked about the vascular events. We know about vasodilation. We know about increased permeability. Those are the two things that you need to remember. Just have those in mind. Then the explanation, you can come up with anything as long as it's scientifically correct. Then that's not a problem. Okay, so when it comes to cellular events, as we can see on this first diagram here, I'm sure you're able to see that the diagram has been cut into 4 parts. So the first part of the diagram, part 1, we're able to see that we have cells which are moving in the middle or at the middle of this vessel. Okay, and this is what I was saying. So when blood is moving, the cells won't move close to the endothelial wall. The cells will be at the middle. Okay, so what will happen is that when these cells, they're at the site of inflammation, the first thing that will happen is called margination and pavementing. Margination is the movement of cells from the center of the center of the vessel to spread out to the corners. Okay, so they'll move from the center, they'll start to spread out. So once they spread out, these cells, they'll start rolling on the endothelial surface. Okay, so after they start rolling, when you have like a lot of cells which have lined up on the endothelial wall, they are rolling. That's called pavementing. Okay, now rolling, these cells do not need to bypass the site of inflammation. Let's say the site of inflammation is right there in diagram 4. So when they are rolling, these cells, they need to be some chemical mediators to make them adhere to the endothelial cell. Okay, to the endothelial wall of that vessel. So once they adhere, the next thing is going to be emigration and diapedesis. So diapedesis is basically the squeezing out of the cell through the endothelial wall, between the endothelial wall. So this cell will squeeze out into the interstitium. Once it's in the interstitium, what's going to happen is that cell needs to migrate, or rather the term being used is immigration from the site of from the site where the vessel is to the site of injury. And that movement from the vessel to the site of injury is called chemotaxis. Okay, the cells are going to be attracted to the toxins which are being produced by inflammatory cells. There are going to be some other inflammatory cells, and there is also going to be toxins which are going to be produced by the microbe agent. Okay, so 4 things happen during cellular events. Let's look at them in detail. Number 1, it's called margination, which is a crucial step, it's a crucial step in the process of inflammation, which involves the movement of neutrophils. Okay, so always remember, when you're talking about acute inflammation, your statement should include the neutrophil. So it's the movement of neutrophils from the center of the blood vessel towards the vessel wall. Okay, so this prepares them for subsequent steps in inflammatory response. So this process of rolling of leukocytes is arrested by special molecules that tightly attach. Okay, so now we're talking about adhesion. So adhesion is driven by two chemical mediators or molecules, which include intercellular adhesion molecule 1, okay, ICAM1, and vascular cell adhesion molecule 1. Okay, so it should be easy to remember because they both have adhesion molecule. So you just replace the beginning. So we have VCAM1 and then we also have ICAM1. So when you are explaining your answer, you have been given to explain the cellular events, then you have been given, let's say, 15 marks. So make sure that you understand that this question will require detail, and that detail you have to make sure that these mediators do not miss. Okay, so you find out that you can write the whole page, you get a 10, but someone who has written in note form has gotten everything because you missed out the mediators. Okay, so take note of the mediators, always mention them when you're writing an essay. Okay, so these two molecules will attach to the leukocyte and integrins of the underlying basement membranes. Okay, so all we are saying here is that the ICAM1 and the VCAM1, these guys, they attach to the leukocytes, then they adhere those leukocytes to the endothelial wall at the point where we need to exit. Okay, so that's margination. Basically, that's margination, then followed by adhesion. So the other point was included here. I hope someone won't miss it. Okay, so the third point is emigration and diapedesis. So before a cell moves to where the site of injury is, the first thing that's going to happen is diapedesis. And diapedesis, basically, like we have said, involves the migration of leukocytes through the endothelial cell lining of the blood vessels. Okay, so the cell will squeeze through the endothelial lining into the interstitium. Okay, so mechanism, it typically occurs through the intercellular junction. Okay, so between two cells, allowing leukocytes to transverse the endothelial barrier. The process of diapedesis is mediated by platelet endothelial adhesion, molecule 1. Okay, so we also have this mediator as well. So take note of the mediators. Take note of the mediators, please, once more. Okay, so after pediasis, the next thing is migration or emigration, which is basically the movement of those cells to the site of injury via chemotaxis. Okay, of which chemotaxis is just is mediated by inflammatory mediators such as C5a, C3a. Okay, so these are pro-inflammatory mediators, as you are going to see when you're dealing with immunology. So C5a, C3a, as well as leukotriene B4. Okay, so for leukotriene, if you want, you can just mention it's mediated by leukotrienes, it's fine. But for these guys, make sure you explain that you're talking about C5a and C3a. Okay, they are anaphylotoxins. So that A stands for anaphylotoxins. So you are going to understand these mediators better again when you're talking about immunology, because you will be talking about these all day. Okay, so the microbes are going to form a chemical gradient. So like we can see in osmosis, it's a movement of water from a region of low concentration to a region of high concentration. So what happens under chemotaxis is that the toxin which has been produced by those microbes, they'll attract our leukocytes to that site. So basically, that process of attraction is what we are saying that it is chemotaxis. Okay, so once those cells reach at that site of injury, they have to phagocytosize those microbes, and we know what phagocytosis is. It's also known as cell eating, where the cell has to engulf, then it has to release some digestive organs to degrade that engulfed microbe or that engulfed material. Okay, so the process of phagocytosis involves three main stages, which include antigen identification and attachment. So each microbe or each toxin which is going to invade your body, it contains a particular portion which is known as an antigen. And this antigen is a part which can be recognized by your antibody. After it has been recognized by your antibody, the antibody will attach to that antigen, then it will trigger some responses there. It's quite a long response which is triggered to depending on the type of response. Okay, so again, yes, immunology. So pathology just focus on the disease. Antigen identification and attachment involves so attachment of the antibody to the antigen special molecule called opsonins. Okay, so this is called opsonization. That antigen, again, it contains a particular portion called an opsin, which attaches to the antibody, and that process of recognition is called opsonization. So opsonins, they are just molecules that have receptors for specific antigen. So this means that each antigen has a specific type of opsonin. Okay, and each type of antibody has a specific type of antigen it can bind to. So these things are specific. It's not just random that, oh, you see that invading microbe, then you attack. No, it has to be specific. So we're saying that because it's specific, therefore are able to detect the presence of any antigen in the interstitium or circulatory system. So they then attach to these antigens to the cell membrane of the neutrophils and other phagocytic cells. There are three types of opsonins. So as you are going to look into immunology, you're going to find out that the type of opsonins, we have immunoglobulin G, okay, or IgG, we have collectins, then we have C3b and C3bi. Okay, so remember we had C5a, we had C5a and C3a. So those, they are anaphylatoxins. Okay, they are involved in chemotaxis. C3b and C3bi, these guys, they are opsonins. Okay, so please have a distinct difference. Do not mix up these things. Okay, so after you have formed an attachment, the next thing is engulfing. And the process of engulfing of an antigen, it involves the presence of an antigen on the membrane of that specific neutrophil together with that opsonin. So this stimulates a special enzyme called phospholipase C. Okay, now the function of phospholipase C, the function of phospholipase C is to hydrolyze the phospholipid found in the plasma membrane. So once we hydrolyze that phospholipid bilayer, it means that that cell, the neutrophil or whatever type of phagocytic cell we're talking about, it can now engulf, okay, can now eat up whatever is on the cell surface. So we're saying that the phospholipase C hydrolyze the phospholipids found in the plasma membrane called phosphatidyl inositol diphosphate. Okay, sorry about that pronunciation, but someone can do it better out there. Okay, so PIP2 into, so it will phospholize that content of that phospholipid bilayer into DAG or dicycloglycerol, okay, and inositol triphosphate 3, okay, IP3. So for these, I would say it's not important to know the phospholipid inositol diphosphate. No, just know that the PLC is going to hydrolyze the phospholipid bilayer. No one will ask you about these two. No one will ask you about no one will ask you about the PIP2 or the DAG, not even in multiple choice, unless you want to include it in your S or something. But yeah, okay. So the inositol phosphate 3 stimulates the release of calcium from its stores into the cytoplasm, which will necessitate for the movement of leukocyte cytoplasm by the assembly of cytoskeleton, forming finger-like projections called pseudopodia. Okay, so now we know how these pseudopodia are coming about, how that pseudopodia of the neutrophil of any phagocytic cell will form. So first, we need to attach to the opsin, which is also attached to the antigen. Then after that, that antigen will trigger or stimulate the release of a special enzyme which is called PLC, of which PLC is going to hydrolyze the PIP2 into DAG and inositol phosphate. So inositol phosphate is very important because it's going to lead to the release of calcium, of which this release of calcium is going to lead to changes of projection of that cell, leading to what we call pseudopodia. Okay, I hope the statement was clear enough for someone to get. So the next thing that's going to happen after we have engulfed that microbe is degradation of the microbe, or rather to neutralize that microbe. So after the antigen is inside the cytoplasm, the neutrophil is going to degrade that microbe in the following ways. So number one, the neutrophil will release special enzymes called defensins. So this part, it's a must know. So defensins, they are just enzymes which create pores in the microbe. Okay, it will create pores in the microbe. It does so to enable the movement of water into that microbe. We know that if there's a lot of water into a cell, the cell is going to burst, and that's exactly its function. So the defensins, they're going to create pores. Okay, they're going to perforate the microorganism cell membrane to cause influx of water into that microorganism, causing cell lysis. Then apart from that, the neutrophils have special enzymes again, which are called NADPH oxidase, of which they tend to stimulate, of which tends to be stimulated by the presence of antigen in its cytoplasm. So when this NADPH oxidase has been activated, okay, when it's activated, the demand for oxygen is going to increase, of which some of the oxygen will be going to act spontaneously with hydrogen molecules to form hydrogen peroxide, and we know that hydrogen peroxide is going to be deadly to bacteria, or bacteria are going to die. Okay, so simply you can try to summarize this. For example, you can say if NADPH has been released, NADPH will lead to increased requirement of oxygen by a cell, of which some of this oxygen is going to react with hydrogen molecule to form hydrogen peroxide, and hydrogen peroxide is going to be deadly to the bacterial cell. Okay, so just try to summarize these things. So you can write all this or you can summarize, but you're going to find out that the marks, they're going to be same. Okay. Question 3. Leukocytes move into the tissue from the vascular space or extravasation through anyone to answer this one. Let's just say best answer. What are we going for? What are we going for? What's the answer? People are quiet. Okay. Is it A, by the action of actin and myosin, or B, predominantly as monocytes on the first day post injury, or C, in response to C3b, or D, in increase to FC fragments of IgG, or E, largely? Okay. Anyway, the answer is there. Someone will read the reasons. Okay. So some other review questions are here. You can try out these questions. Yeah. So if there's any question, you can ask. If there's no question, we end here for part one of inflammation. Any question? Okay, I'll take it.